when I say cell, we're talking about bacteria. When we talk about microbial or bacterial genetics, there is certain things that we should actually be aware of: the genetic characteristic of the bacteria and their changes, and the role of these genes in the clinical setting. So we're talking about transfer of the gene and how the gene will actually transfer different characters to, a second generation or from a generation to another generation. Okay, can you recall from your second lecture? So if I ask you bacterial genetics, so what's the genetic elements of the, let us say, eukaryotics? And when I say eukaryotics, we mean bacteria. So can you recall what are the genetic, elements of the bacteria? The chromosomes, exactly. Plasmid. Okay, so can you tell me what the difference between chromosome and plasmid? This, the most important difference, because they share, yes?
Plasmids are not in the nucleoid.
They are not in the nucleoid.
They are extra chromosomal.
They are extra chromosomal, okay? What else?
It is the characteristic you want to transfer it to another.
Exactly, nice. So the plasmid actually play a major role when we talk about, transfer of specific functions. Okay, all right, we're doing good. So the two components of the, or the main components, because we're going to talk about other stuff in genetic, bacterial genetics, is the chromosome. And if I just look on this, it's circular, double-strand DNA, have essential genes for replication and metabolism. Why I mentioned that? Can plasmid play a role in the bacterial growth? No, great. So they are haploid. That means you have only one copy of each gene, and the chromosome is a single in each, bacteria. Okay. When we talk about plasmid is an extra chromosomal material or extra chromosomal gene. They are smaller in size compared to chromosome. So this is like a difference. They replicate autonomously. Remember, chromosome just give a copy for one single. So plasmid could be like single or multiple. The plasmid, as your colleague was saying, they are incorporated in the chromosomal of, bacteria. So in themselves chromosome, and when they incorporated in the chromosome, we call them episomes. Okay? And of course, they play an important role in the genetic engineering because they actually transfer genetic informations. Okay. The plasmid, based on their, properties, classified into two. The first one is conjugative. Where did we talk about conjugative before, or conjugation? Yes. We mentioned when we talk about bacterial structure, we said that bacteria have, appendage in the surface. They play a role either in attachment, movement, but we also mentioned they could play a role being asexual, bar to connect the two cells together and transfer plasmid from one cell to another. Okay, so this is exactly the conjugative plasmid. So those are the plasmid that could promote transfer of the plasmid from one cell. The cell that gives the plasmid, we call it what? Donor. The one that receives, we call it recipient. Are we clear? Okay. So these conjugative, as we just discussed, they have the villi, and the villi will help in the process of conjugation. So conjugation is one of the gene transfer mechanisms, and we're going to talk about other. All right. We also have non-conjugated plasmid. Those are the lazy guys. So they sit doing nothing. They don't have any function. So conjugated and non-conjugated. Conjugated responsible for, promote specific function. But there is also other. If we think of the classification based on the function, we have fertility plasmid from the name. So they are responsible for, initiation of conjugation, which we just mentioned. We have the cold plasmid that produce special protein, and this protein actually responsible for killing other bacteria. We have the degradative one. From the name, degradative mean that they actually do digestions of the organic compound and other unusual substance. And the last thing is the valence plasmid. And all right, I'm not going to go through this a lot, but I just want to ask you, you know that the DNA structure is built of deoxyribonucleic acid, true? And the difference between DNA and RNA is what?
The strand is the nucleotide sequence.
The nucleotide.
Yeah.
So uracil is replacing thymidine. Okay, and what else? Okay, in the sugar, the ribosome in the RNA, ribose in the RNA, and then deoxyribose in. Okay, great. We're going from here. The most important here, I would like to take a note from this slide that genetic information will contain within the cell genome. And then we say cell genome. What do we mean by genome? Like when I say like a genome, it's a sequence of DNA.
The entire complement.
The entire complement. So it's the genes arranged together. Collectively, they make the genomes of the bacteria. All right. Okay. So this is just like